It's my pleasure to introduce Dr. Natalia Egorova, who has until recently was working at the Florey. She's now University of Melbourne. Many of you know Natalia. She's currently an ARC DECRA Fellow at the University of Melbourne, and she's still honorary with us at the Florey. Natalia has had a really stellar history in the sense she was interested initially in language and linguistics, and then she did her PhD and then did postdoc work in pain, and now is interested and then became interested in stroke. So she has an incredibly broad expertise as well as superb imaging skills that she is bringing to her data, and she has a very beautiful little baby too. Those pictures, those most recent pictures. Oh, she's so cute in that hat. She's got wide-ranging research interests, and today she's going to speak about some analyses from Canvas, which we're really excited about, looking at advanced imaging using diffusion, but not DTI, but looking at a Fixel-based approach to white matter degeneration after ischemic stroke.
Thank you, Amy. So I'm going to talk to you about white matter and neurodegeneration, but I'll take you on a circuitous route and also talk about grey matter and function. So this diagram shows the timeline of the different processes that happen after stroke, neurodegenerative and restorative. And roughly we can talk about focal and remote and global processes, and I'm going to just briefly mention them in function, grey matter and white matter. So when there is a focal lesion, say in the motor cortex, you just lost the motor control over that particular area, so that's fairly clear. But there are also remote functional problems. For example, you get a stroke in the motor cortex, but you then get the whole shutdown and the contralesional cerebellum doesn't work properly. So the things that are connected are also affected by stroke. Finally, you can get global effects. This is at 3 months, a reduction in default mode network connectivity in stroke patients compared to controls, and there are no stroke exactly in the network. So this is just a global effect of stroke on the brain. Similarly, we can look at grey matter and, of course, talk about the lesion and the infarction and the penumbra, but in addition, we can also observe remote degeneration, say in the hippocampus, or here you can see the thalamus degenerating from 1 day to 1 year, ipsilegionally mostly, but also contralesionally. Finally, if we did a whole brain analysis, say with VBM, we could observe brain shrinking across the whole brain. So there is something that stroke does to the brain that causes further neurodegeneration. So what about white matter? We definitely not take into account that white matter also is very susceptible to ischemia. And unlike the rodent brains that are only 14% white matter, human brains are 50%. So in the same lesion, you get a lot of white matter disruption as well as the grey matter. In addition, there are numerous accounts of this classical Wallerian degeneration of the cortical spinal tract. Here you see 1 month and 3 months post-stroke. The lesion gets smaller, but so does the cortical spinal tract, not only ipsilegionally, but also contralesionally. Interestingly, despite the fact that we see focal and remote and global effects everywhere, in white matter there haven't been a lot of accounts of that. There is one study in very acute stroke, 48 hours post-stroke, and they looked at contralesional hemisphere in normally appearing white matter, only in one hemisphere, so it's not quite global, and show that indeed there is white matter problems after stroke compared to controls, but we don't know whether it's the effect that was prior to stroke or it's the effect of stroke, but it could dissolve and resolve on its own in a couple of weeks. So it would be interesting to know what happens longitudinally and potentially at a later stage. So one very recent study, in fact, from Andrew's group, did that, but found no global effect. So this is fractional anisotropy map in stroke 1 to 3 months longitudinally, and no effect, global effect. However, they did find ipsilegional motor cortex effects if there was an AHSS. So they found some remote degeneration, again, some evidence of white matter degeneration, but not globally. So perhaps it could be the sample size problem or the methods limitations, or in all of those studies there could be diffusion quality, imaging data quality problems. So any of those things, we just wanted to see, is there a global white matter degeneration after stroke in our study? And we had access to the Canvas study at 3 months. We were lucky to have many subjects, 104 stroke patients and 40 controls, matched on age, sex, and intracranial volume. There were differences in education, and we know that, so we control for that usually. The stroke cohort was fairly mild strokes, and the lesion volumes were also pretty small. You'll also notice that there were more right hemispheric strokes than left hemispheric strokes. So we also had very good quality of the imaging data, 60 directions with the b-value of 3000. And that allows us to do a Fixel-based analysis as opposed to the traditional diffusion tensor imaging. So in diffusion tensor imaging, you essentially take the brain, chop it up into blocks of about 3 millimeters, and then average whatever values of diffusion you're looking at within the voxel. However, you can imagine that there are lots of axons going through a 3 millimeter block, and also that some of the fibers could be crossing, producing as a result conflicting values and potentially diminishing the power, or even producing nonsensical results. So the Fixel-based analysis takes into account the directions of the particular fibers, and as a result you get the metrics within fiber and not within voxel. And we can look at fiber density, so rarefication of the axons. You can get cross-section difference or the combination of the two, reduced fiber density and cross-section. So in all of the results that I'm going to show you, I'm going to display decrease in fiber integrity in stroke compared to controls, and there will be we account for age, total intracranial volume, sex, and education, and all the results are significant at a very strict family-wise error-corrected p0.05, and you will see that the results are shown by fiber orientation or the effect size. This is the relative to control. So going straight to the results, in fiber density, and this is a reminder of what it means, we have pretty much the whole brain, all the white matter tracts showing decline, and this is up to 50% decline. You will appreciate the corpus callosum differences, the association fibers like in inferior longitudinal fasciculus. Note that yellow means more of an effect, more of a reduction, and it's more pronounced on the right compared to the left. So if we look at the fiber cross-section, that's when the fiber is shrinking, you'll notice massive decline in the superior longitudinal fasciculus bilaterally, but more again on the right. And finally, if we look at the combination of the two, you'll see pretty much the combination of those two, but this time only the right superior longitudinal fasciculus is significant. So is this because we have a right hemispheric cohort predominantly, or is it something that's global and characterizes a neurodegeneration after stroke? So when we split our subjects into left hemispheric and right hemispheric, and you can appreciate that these are just pretty much mirrored images of each other with just different numbers of subjects, and we compare left versus control or right versus control. The results look somewhat different, but remember that this is only 40 subjects and we are correcting for quite a lot of covariates. If we strip down the covariates down to age and total intracranial volume, which is typically used in Fixel-based analysis, we'll actually recover all of those bilateral global connections in the splenium here, and we also see that on the right there is a disruption of the right corticospinal tract, and on the left there is a disruption of the left corticospinal tract. Interestingly, the right superior longitudinal fasciculus here is on the right, just like what I showed you earlier. So this suggests that there are lesion-related and also global effects. So going back to this chart, we get the focal lesion, that's somewhat expected. We have functional remote and structural remote changes, and our data shows that as well. And then we have global functional and structural changes, and previously we saw very early effects of stroke on white matter. And now we put the missing puzzle here as well. There are global effects of neurodegeneration in white matter. So that answers our question, but it doesn't answer all the questions, because being in the vascular neurodegeneration symposium, obviously there could be something that precedes ischemia. So there's a lot to look at the association between, say, white matter hyperintensities and the results that we've got. So lots of future directions. This is by no means a finished project. We'll look at white matter hyperintensities. We'll consider cognitive effects. For example, here I'm showing you only a region of interest, analysis in one of the study showing that corticospinal tract is associated with motor effect problems and the superior longitudinal fasciculus with the cognition. So we could definitely look into that, given the superior longitudinal fasciculus finding here. We can also, previously the hypertension study showed that there was a link between the SLF and the frontoparietal network and the executive function. So since we are seeing the differences again in SLF, and it's usually associated with attention. And we have previously shown, Michelle Feldman showed a difference in the dorsal attentional network. So obviously we have these bits already here and here. So it would be nice to join them up and see how they are related. So with this, I'll stop and open for questions, but there are more questions that we can ask still at this stage than we can answer. So thank you very much.
How amazing are these data? I mean, it's just extraordinary. This is something that Natalia's really been only doing the last couple of months, and I think they really represent, for me at least, that intersection between the advances in the methods. We've got better sequences and better methods. Really, a collaboration with Alan Connolly's group and Shauna Farquharson being able to give us this extraordinary data. So, thank you.